and the Center for Healthcare Innovation. I'm going to show you just a couple of slides, and then we're going to get going, and I'll tell you how this is going to work. If you hopefully can see my screen here, a slide on why we're doing this. So we're really trying to highlight the creative and innovative work going on here in the HVC in the section of cardiovascular medicine. We're trying to reward those who are creating value and building a culture of innovation and provide recognition and support. We want to stimulate discussion. We want to provide a roadmap and practical tools, and we want to have fun most of all. So it's really wonderful that we have 8 faculty and fellows who are present and house staff who are presenting here. It's really amazing. I'm blown away by the level of interest and the participation. This is a 2-part grand rounds, December 1st today, and then December 8th next week, where we'll have talks on practical guides to innovation by Margaret Cartiera and Michael Singer. And I want to offer special thanks here to Eric Velasquez, Margaret Cartiera, and Joe Bison for really supporting these efforts, and really special thanks to Liz Reitman, whose organizational skills and engagement are actually central to getting this whole thing going. I'll just introduce the judges. Eric Velasquez is the chief of section of cardiovascular diseases, obviously, and physician in chief of Heart and Vascular Center. Of course, we all know Dr. Velasquez, and we're privileged to have him here providing his insights for this competition. Michael Singer, who's a longtime friend and colleague of mine, who's an entrepreneur and who was a Yale medical student, also got his PhD from Yale, and went to Boston, trained at Brigham and Mass Eye and Ear, and became an inventor and an entrepreneur, and has met with some success along the way, and is going to provide his insights to us. John Soderstrom, who's the managing director of OCR, who many of you know, and provides leadership and vision for that organization and across the university. And Margaret Cartiera, who some of you may know and some of you may not, but hopefully all of you will, who's the director for the Yale Center for Biomedical Innovation and Technology, CBIT, and the Sci Center and the Center for Healthcare Innovation. She's the innovation director, and she's just a fabulous person, and really the brains and the brawn behind a lot of this stuff, both. At the end, I will ask the audience, if you'd like to participate in ranking the presentations. I'm not going to show the rankings publicly. I think that would be counterproductive. But if you do go ahead and do these rankings, you can go to pollev.com, Daniel Jacoby 170, and provide rankings according to the presenters. This is in no particular order. It's just the order of the presentations, which is not specific to anything. And publicly, even next week. So it's going to be private. The rankings will be private of the audience, but for your own participation, if you're interested, you can participate in that. And I'll provide that to the judges. Dr. Churchwell was going to be one of the judges today, but he had to be out at the last minute because he has to do a presentation to the board of directors for Yale New Haven Hospital, which seems an important conflict and one that we can all understand. With that, I'll stop sharing my screen, and I'll invite Dr. Elefteriades to show his slides and be the first presenter. Congratulations, Dr. Elefteriades, and thank you.
Thank you, Dan. Can you see my screen?
Yes.
Okay, I'd like to present our brain cooling catheter, which is designed to provide brain protection in subarachnoid hemorrhage, with likely applications also for traumatic brain injury and stroke. And the means of protection is intracranial cold. It was Bigelow in 1950 who made the cardinal recognition that the metabolic rate of the brain decreases exponentially as the temperature falls. We apply this every day in the operating room when we replace the aortic arch under total hypothermic circulatory arrest. And this has captured the interest of the general public, and we've had many TV shows and magazines come and film this because it fascinates the public that the brain can be protected with no blood flow. I could show you a bunch of graphs and charts from our publications about the effectiveness of cold, but instead I'd like to show you this little 20-second video. This woman is 78 years old. The day before this video, We replaced her arch. She had 49 minutes of no flow to her brain at 18 degrees centigrade. My name is pretty hard to spell, but check this out.
Spell your doctor's name forward. Elefteriades. And reverse. Sedeiretesele.
So I think that's a pretty good demonstration of the effectiveness of cold in protecting the brain. Now, we started out by protecting the spinal cord against the devastating injury of paraplegia after descending aortic surgery. We developed this catheter that recirculates a refrigerant through inflow and outflow lumens. It preserves a central lumen for draining spinal fluid to keep the pressure in the canal low and encourage forward blood flow. And this was supported by National Science Foundation Phase 1, Phase 2, and Phase 2 supplemental grants. These experiments were successful, and this technology was acquired by CryoLife, and this is the commercial version that should be at the bedside pretty soon. Now, I'd like to move on to the current topic, and that is protecting the brain by putting one of these catheters with a balloon at the tip directly into the lateral ventricles of the brain. As you can see, this is very effective at bringing down the brain temperature. We did these experiments in sheep, and histology showed no evidence whatsoever of any brain injury from the cold itself. Now, in subarachnoid hemorrhage, even after the aneurysm is controlled, there is tremendous incremental injury to the brain from fever, diffuse cerebral vasoconstriction, inflammation, neuronal ischemia, edema, and increased intracranial pressure. Almost always a catheter has to be placed anyway into the ventricle to drain fluid out and bring the pressure down. Systemic hypothermia has been tried and it showed some benefit, but it carries with it a whole cascade of adverse consequences from cooling the whole organism. So the technique of ventriculostomy has been used for hundreds of years. It was used on Abraham Lincoln in 1850. Every neuroresident learns how to do this in his first week, and it's a relatively minor bedside procedure. In subarachnoid hemorrhage, because the catheter has to be placed anyway, we see our brain cooling catheter as providing free hypothermia with no additional difficulties beyond the simple placement of a traditional ventricular drain. You can see here now, we did MRI imaging of the brain temperature in sheep when applying our cooling catheter, and you can see the beautiful propagation of the cooling wave throughout the cerebrum. The rights to the brain cooling catheter are still held by the Yale spinoff that has done the spinal cooling catheter. This was all done with OCR under the direction of John Pusis. So we've had extensive discussions with the FDA. They've asked us to do one final sheep experiment with very long duration cooling, and then they feel they can give us the go-ahead to do the first in-man 10-patient clinical trial in the neuro ICU at Yale. The budget for both phases is $2.5 million. We have a revised grant into the NIH. They invited us to reapply with some revisions, and we're hopeful, but of course you never know about that, and we're of course interested in any additional avenues of funding, which would be welcome. Thank you.
Dr. E.
Thank you for your attention.
Oh, thank you so much, Dr. E. Is that the end of your presentation?
That's the end of my presentation.
Wonderful. We have time for a question from one of the judges.
And maybe I'll go first. First of all, thank John for a wonderful presentation and this work that you've been doing. I think it's impactful and impressive in many ways. So my question is rather simple. If cooling is good, is too much cooling bad? And I guess, do you have evidence to address that? And what have you found when you've taken? What is the is there a threshold beyond which cooling can provide, disorder in brain and potential consequences?
Thank you, Eric. Even a small amount of cooling is beneficial. The drop in metabolic rate is 50% per 6 degrees centigrade. So it's been shown that even a 0.5 to 1 degree cooling is very protective. We can easily get 4 or 5 degrees of diffuse cooling of the brain. that's well within the safe level. Eric, in the operating room, we cool to 18 degrees, from 36 down to 18 degrees, and it's perfectly safe. I've done it 2,000 times myself. To the inflated balloon catheter in the lateral ventricle. So we hope that that won't be a clinical problem.
And maybe a quick follow-up, Dan, if it's okay. What in the catheter, in the structure and the engineering of the catheter, what safeguards have you put in place to prevent the cooling to go below 18 degrees or whatever threshold you'd like to set?
There are tremendous safeguards. That device that I showed you has safeguards against excess pressure, against any rupture of the catheter. It very carefully controls flow rate, and it absolutely controls temperature, and it's all computerized.
Wonderful, Dr. Elefteriades, and thank you, Dr. Vasquez, for those questions. If audience has further questions for Dr. Elefteriades, please keep them, a note of them, and hopefully we'll have time at the end. Or if other judges have questions, hopefully we'll have time at the end. Thank you so much for a fantastic presentation and an interesting device. I'd like to invite Dr. Friedman, Dr. Daniel Friedman, for our next presentation.
All right, can everybody hear me? And can people see my screen?
Yes.
Yes, Dan.
Okay, terrific. All right. Well, thank you, everybody. I'm going to describe a project I've been working on, Jim Freeman, titled Method and System for Determination of the Vector of Ventricular Activation using 3 extracardiac electrodes for rhythm discrimination and localization of VT and PVCs. So the problem, it kind of goes without saying, but it's worth reiterating that the optimal treatment and management of patients with non-transvenous ICDs, and by that currently I mean subcutaneous ICDs is the only system that's FDA approved and available. It relies on accurate discrimination between SVT and VT. In traditional transvenous defibrillators, these devices can use intracardiac near field signals, which allow for much easier differentiation of atrial activity, ventricular activity, and T waves. However, non-transvenous devices must rely exclusively on far field signals, which are much more difficult to analyze, and misidentification of an arrhythmia can lead to morbidity, mortality, or impaired quality of life due to either undertreatment or overtreatment. The Achilles heel of Boston Scientific subcutaneous ICD is the inappropriate shock rate for T-wave oversensing and oversensing of myopotentials. And a pre-procedure screening is required before implantation of these devices. And approximately 10% of patients are not suitable due to the ratio of the QRS complex to the T-wave based on screening. Medtronic has a competing device that is overall similar and will likely have similar issues. Beyond this, optimal management of VT requires an understanding of the site and origin and the number of unique VTs. Treatment for multiple VTs, multiple foci PVCs can be very different than if you have one single recurrent morphology. So let's talk about the opportunity here for improving care. So non-transvenous ICDs, and the Boston Scientific device is what I've got shown right here. As I mentioned, uses far field signals that are derived from one of 3 extracardiac electrode pairs. In the subcutaneous ICD, the electrodes are located in the can at the proximal aspect of the parasternal coil, demarcated by B, and then the third electrode is at the tip of the substernal, or of the parasternal coil, marked with A. And any combination of these vectors can be used for sensing. You use one pair for sensing during the current iteration of the device, and you can see that Boston Scientific has named some of these vectors primary, secondary, or alternate. However, the information from the other 2 electrode pairs is not recorded or analyzed, and from my perspective, this represents information that is wasted. So what's the solution? So I propose, and Jim and I have been working on this, a vector-based approach where far field signals from 3 electrode pairs are transformed into a vector cardiogram that is able to represent depolarization in much more exquisite detail than the standard far field representation from a single electrode pair. This schematic here is actually of Medtronic's extravascular ICD, which is similar to Boston Scientific's design, except Medtronic's design has the coil actually under the sternum and the anterior mediastinum. However, the relationship of the electrode pairs is similar. What I'll draw your attention to here is that the different electrode pairs actually contain information in different planes. So if you see here, the blue arrow indicates information exclusively from the Y plane. The can, which is at the mid-axillary line, when joined with the proximal electrode, can give information to the X and the Z planes. And when you compare that to the electrode pair derived from the can and the electrode at the distal coil, you get information from actually the X, Y, and the Z planes. The figure on the right just demonstrates how these planes lie within the chest. So the rationale for why this project will work. So a vector cardiogram is well known to be able to be derived from a standard 12-lead EKG, and the prognostic value of these derived vector cardiograms has been demonstrated over and over and over by my group and others. What's that?
About 20 seconds, Dan.
Okay. So the first 40 milliseconds of the QRS complex or the vector loop represents septal depolarization. Each patient's septal depolarization can be defined in 3D space using this vector cardiogram, allowing subsequent complexes to be characterized as septal versus non-septal. And by using that septal activation sequence as a fiducial and with an understanding of anatomy, you can define SVT versus VT and localization of the ventricular beat within the heart. So just real quick in terms of the market opportunity, so 157,000 ICDs are implanted annually. About 55% of those patients are considered to be eligible for a subcutaneous device. And by including this type of technology in an existing or developing platform, you can increase the market share of these devices and also improve care of patients by integrating currently unused bits of information. So with that, I will thank everyone for their attention. And just let everybody know that at this point, we're not quite as far along as Dr. Elefteriades. At this point, we're trying to obtain funding for some animal studies. So thank you.
Thanks so much, Dan, and I appreciate your efforts to stay within the time limit. That was great. I'm sorry you had to rush at the end. Dr. Churchwell was assigned as the judge to ask a question of you in his place because it was the last minute. I'm going to take the place of asking you the first question, and that is, if you receive funding to pursue animal studies, what would be the most important data you could obtain from those animal studies, and how would you use that data to pursue the next level of studies or partnership with industry?
Yeah, so the important next step is to actually do an animal study where we would probably put EKG leads in the locations of the electrodes and actually do pacing in different locations to and also induce block in his Purkinje system to look for aberrant conduction. So we can clearly prove that we can, one, define the septal activation sequence in an animal, and two, demonstrate that it's very clearly different in ectopic beats.
And how would you use that to speak with industry partners or develop your project further, that data?
Yeah, so we've been in touch with certain potential industry partners, and everybody is enthusiastic about the idea, but people are just interested in more data. And so, you know, there's a lot of reasons based on prior work why this should work, but what we need is a little bit of data demonstrating that we can successfully take these concepts and transform them into this application. And even if it's a sort of a modest amount of data, I think it'll go very far at sort of igniting these types of partnerships.
Thanks, Dan. That's terrific. Appreciate your efforts, and it's really exciting work that you and Dr. Friedman are working on. It would certainly be great to have fewer shocks, unnecessary shocks for our patients. I'd like to invite Dr. Ghazizadeh for our next presentation. Dan, if you can take down your slide.
Good morning, everyone. I'm Zaniar. I'm one of the third-year medicine residents. I'm very excited to talk about the startup I recently co-founded on using stem cell and machine learning technologies for drug discovery. My co-founders, Dr. Kishinevsky and Dr. Fatai, actually were lab mates at Memorial Sloan Kettering, and their work went into Blue Rock Therapeutics that 2 years later bought out by Bayer for $1 billion. And basically Blue Rock Therapeutics used stem cell technology for cell replacement therapy. But we are planning using stem cell technology augmented by machine learning for drug discovery. Have you ever felt your heart beating out of your chest because you're anxious? That's how I feel today. And that's because of our peripheral nervous system. And that's an example of how body-brain interaction works. Basically, peripheral nervous system regulates our tissue, and that is an underlying cause for many disease, but they have been understudied and we don't have effective treatment for them, especially because we didn't have any high throughput or scalable way of getting access to peripheral nervous system. Our startup focuses on 2 indications, GI disease and cardiac disease. But for the interest of the audience and this session, I'll focus on the cardiac indications of body-brain interaction. There's growing evidence that the damage or stress to our heart signals our brain, and this in turn signals our heart again. And that can result in atrial ventricular remodeling, and that can cause certain types of cardiomyopathies or arrhythmias. And in fact, one of the most common arrhythmias, which is atrial fibrillation, there's growing evidence for that neuromodulation can result in onset or progression of the disease. So how our platform works, basically the Exir engine uses stem cell technology to generate the cell type of interest and uses a target agnostic iPSC-based phenotypic screening, and that is augmented by machine learning. Step 1, like we generate iPSCs from human with certain disease that we are interested in, and then we can differentiate that with appropriate signaling to any cell type of interest, and which is this step. And then we study their phenotype in vitro, and we run a high throughput screening to identify compounds that they reverse the phenotype. And this is the next level that makes it unique, that like then we feed this data from this assay or like multiple assays that we do on these cells, we feed it to our machine learning platform that runs through millions and millions of compounds, and that can come up with new chemical entities that can reverse the phenotype. Then we study those compounds and then we test them back on our human in vitro models or in vivo models of the disease, and then we can select them for preclinical studies. In the next 2 slides, I'm going to talk about a couple of proof of concepts about this platform, and they are available in the co-founders publication. So on the left, I show a schematic that how we can turn iPSCs into autonomic neurons, and these autonomic neurons can secrete like neurotransmitters, and we can get iPSCs from disease or healthy and generate autonomic neurons, and then study and do high throughput screening and identify what can increase or decrease these neurotransmitters. For example, we have data for nitric oxide, we have data for epinephrine, which are important in pathologies and physiology of the cardiac and GI system. On the right, I show you a couple of immunofluorescence and how we can generate atrial cells from iPSCs from either healthy individuals or individuals with atrial fibrillation and study their structural proteins. In this slide, I'm going to show you how machine learning plays a role. So here, this is a recent study that we published in Cell Stem Cell that showed that we used iPSC-derived ventricular myocyte. We ran in vitro high throughput screening to identify compounds that can modulate a receptor on the ventricular myocyte. Then we gave this data to our machine learning engine, and this, based on the algorithms, came up with new chemical entities. And we tested back on our human cells. And this showed that, like here in purple, are the in vitro generated compound, in vitro identified compounds that like they could reduce the level of the receptor, which we were interested in. And in red, these are the machine learning generated compound, new chemical entities, and they were even more efficacious.
20 seconds, Dr. Ghazizadeh.
Thank you. So the unique advantage of our platform is that we can study many cell types together. So there are other companies that use CNS or cardiac cells to study them individually, but we are actually looking at cell-to-cell interaction. We are looking for $30 million in funding to generate the platform and study the already identified hits in preclinical models, and which are the GI platform and the cardiac indications, gastroparesis and AFib, but we are planning on expanding cardiac indication to HFpEF and cardio-oncology programs. With that, I want to thank the audience for their attention and some publications of the co-founders.
Fantastic. Thank you so much. Wonderful presentation. We have a question from one of our judges.
Yes, hi. Could you say more about your business model? How do you intend to get paid?
Basically, there are two different ways that we're thinking about doing it. One of them is a partnership with pharma, potentially, that we identify hits and then partner with pharma that is interested in the disease. For example, one of our co-founders has already connections with Takeda for GI indications. That's one way of doing it. The other one is that's just after identifying the hits. But for the other one, like we identify the hits, we do all the preclinical models, and then at the end, again, partnering with pharma for like potentially marketing. Those are the two different models that we're thinking about. Thank you.
Thanks so much, and thanks, Dr. Singer. We'll go ahead, Dr. Ghazizadeh, that was a wonderful presentation, really exciting work. We're going to go ahead and move on to our next pitch. To allow everyone adequate time. And I would like to welcome Dr. Mani.
Okay, thank you for the opportunity. Everybody sees my slides? Yes, so today I'm going to talk about an incretin-like peptide that is called SELA2A, which is a novel peptide for the treatment of metabolic syndrome. And metabolic syndrome is the fastest growing risk factor for diabetes and atherosclerosis. And insulin resistance is the central factor among all the metabolic syndrome traits. The mechanism, however, underlying the transition from insulin resistant to development of pancreatic failure, beta cell defect, and diabetes is not understood. And this is an impediment for the drug development for metabolic syndrome and type 2 diabetes. So we use the genetic approach to identify a pancreatic enzyme that actually regulates insulin secretion and insulin sensitivity, and its defect links two traits of insulin resistance with diabetes. We first identify large and smaller families who were characterized by myocardial infarction and coronary artery disease under the age 30. They had diabetes type 2 or were insulin resistant and had most of the metabolic syndrome traits, and use their linkage analysis to identify mutations in a pancreatic chymotrypsin elastase that was mutated in all these families, and the substituted amino acids were actually within the very highly conserved region of the encoded peptide. So SELA2A mutations actually impair the catalytic function of these serine protease. For example, the aspartic acid 121 substitution with asparagine 121 disturb the catalytic triads, which consists of serine, histidine, aspartic acid, which is required for activation of serine to cleave the peptide bond. Almost all of these mutations dramatically reduce the elastase activity, close to actually zero compared to the wild type protein. SELA2A was also found to be ubiquitously expressed, not only in the exocrine pancreas, but also in other tissues, as shown here in mice, in mice mRNA and protein, in many other organs of the mice, and in human by immunohistochemistry in pancreas, adrenal gland, the small intestine, and large intestine, and interestingly, within the pancreatic islets itself, within alpha and beta cells. But most exciting was that actually to identify it as a circulating peptide. Interestingly, patient with V121 end in the larger family with the V121 substitution had a very high levels of SELA2A, but reduced activity. And when we actually fed normal individuals, SELA2A rose in parallel with insulin and C-peptide, and in a very, very perfect way, so with an R almost close to 1. When we actually examine patient with obesity, with hyperglycemic clamp and oral glucose tolerance tests, again, we noticed that the insulin and SELA2A levels may rise in parallel with each other. To understand if SELA2A triggers insulin secretion, we injected LDL receptor knockout mouse who were diabetic with recombinant SELA2A And we noticed dramatic reduction in the glucose levels, which was associated with a rise in the C-peptide and insulin. Interestingly, SELA2A had no effect in wild-type mouse, which had normal glucose, suggesting safety of this drug in the treatment of diabetes. To understand that if this is a direct effect of the SELA2A on pancreas secretion, we treated the isolated rat pancreatic islets, and we saw actually elevated levels of the insulin in response to the wild type, but not a mutant SELA2A. And this was associated with a rise in transient, calcium transient, which was actually much reduced in the mutant SELA2A. And this was caused by cleavage of the calcium channel, which activated the calcium signaling. Most excitingly, actually, SELA2A activated insulin signaling. Here shown by AKT phosphorylation, SELA2A wild type, in the absence of insulin, activates AKT and downstream pathway. So it's also a sensitizer for the insulin function. SELA2A also attenuates platelet activation. Integrin alpha 2B contains alanine proline, which is a motif, and this is a component of the 2B3A, which this enzyme cleaves and therefore inactivates the 2B3A and results in actually lower aggregation of the platelets, as shown here in this figure, and also in the micrograph, as you see here, that the mutant SELA2A causes aggregation, as the mutation in the patients does, but the wild type actually reduces the aggregation of the platelets. So what I suggest is actually we want a funding for safety of this drug, and we have generated a knockout mouse model, and it's perfectly a perfect model because the knockout mouse model on chow diet has insulin resistance, and when we feed them, actually they develop. I don't know why my slides are not moving, unfortunately. For some reason, okay. So the mouse actually on chow diet develop insulin resistance, and on the high-fat diet, they're actually hypoinsulinemic. So the best model that one can study the link between insulin resistance and hypoinsulinemia. And mouse actually, when they are deficient for this gene, develop fatty liver and have a high TG secretion. So we would like to have a funding that actually we can study these mice and also see the safety of this drug for future treatment. What I actually showed you is, in summary, shown here, that wild-type SELA2A is an incretin-like peptide that inhibits platelet apoptosis and aggregation, but also regulates insulin secretion and insulin sensitivity, and maintains normal glycemia, normal insulinemia, and normal state. So thank you. I just want to mention this. People actually were involved in this study, which was a collaborative study.
That is amazing. Wonderful, Dr. Mani. Thank you so much. We're a bit short for question time, so a very brief question, please.
Are you kidding me? This guy just gave me a freaking tutorial. It'd take an hour to really dissect. But quick question for you. When you looked at the mice, what adverse effects did you see from the, I saw that the insulin dependence go down, but from the other, because this seems to be ubiquitous in other tissues, did you see any damage in other tissues or any effects in any other tissues in the mice?
Well, so what we looked at that time was just the wild-type mouse treated. LDL receptor knockout mouse treated. And
oh, okay. So you haven't actually. Okay.
But no, but in the knockout mouse, actually, we don't see any problem. Mice that actually are deficient still survive, but obviously they are insulin resistant. And they also have increased neutrophil counts in the tissues and in the blood. But what we are suggesting that actually SELA2A itself could be a treatment, and its loss is causing disease. And we want to see, when we treat this SELA2A, do we see any long-term side effect? So we actually have generated the pump. We're going to inject long-term and see if we can actually, for example, use it in a diabetic mouse model, treat the diabetes, and also if this can be independent of insulin. So we have other models to study that.
That's exactly what I wanted to know. Thanks.
Thank you so much. Thanks, thanks, John. That was one very hard to ask a question with such a detailed scientific presentation. Thanks, Dr. Mani. Wonderful work as always. Thank you for participating. I'd like to ask our next contestant, our next participant to present, which would be Makoto Mori. And I think all the audience, as a word of thing, I think all the audience and the participants can see how challenging it is to present an idea in such a short timeline. And I want to, in the middle of all this, just again, congratulate our PitchFest participants for doing that. It's a real challenge to do it, and it's wonderful work that everybody has done so far. Thank you.
So Dan, I have to add that when you asked me to remove the voice from the slides were kept moving themselves. So that's why the problem was during the presentation.
Oh yeah, I'll explain at the end about that. But yes, thanks, Dr. Mani. Appreciate that. Go ahead, Dr. Mori.
Good morning. Sorry, I'm on mute. Oh, sorry. Good morning. I'd like to present a screening algorithm for thoracic aortic aneurysms that is embedded in an app platform. And this is the team with 2 attending cardiac surgeons, as well as a PhD biostatistician and the app developer who's been helping us with budgeting, as well as a mock-up of an app interface. So the problem that we would like to address through this tool is that under detection of thoracic aortic aneurysm, which is a major risk factor for aortic dissection, it is an issue because the aortic dissection has a high case fatality rate of over 20% within the 48 hours of onset. Aortic aneurysm is a major risk factor. However, it is a modifiable one through preemptive identification and also prophylactic aortic operation to replace the aneurysmal segment of the aorta. The problem that we are currently facing, though, is that aneurysm is asymptomatic, and there is no established screening protocol in thoracic aorta, unlike that of the abdominal aortic aneurysm. So the ideal framework is there is a screening to identify patients at increased risk of having aneurysm, undergo surveillance, and at the right timing when the size increases to an operative indication, there is a preventive surgical intervention. However, the screening process is compromised because we have limited understanding of the risk factor as well as a tool to quantify the patient's risk. So consequently, we are performing more of these prophylactic operations, but we are observing increasing in the incidence of aortic dissection. So at the public health perspective, we are really not doing a great job at minimizing this disease burden. So the solution that we are bringing to the table is this risk prediction algorithm that is embedded in an app platform. The unique feature of it is that it is derived from a fairly large data set of 21,000 CT scans done at Yale, of which 2.6% had aneurysms. Really, the distinguished feature from other large data set is that this is all manually remeasured with a standardized technique. And that's important because claims data set falls short in distinguishing ascending from descending thoracic pathology, and data sets like EHR-based data encounters issues where the radiologists are not using a standardized techniques or are using different size threshold to call what is aneurysmal. So we believe this is a very unique defining feature of this data set and the algorithm that is derived from it. So we developed a risk model based on a simple demographic and comorbidity predictors because this does not require any advanced testing to get further predictors. Any patient having clinical encounters would have these variables, so allows for wide-scale implementation. So then this is an instrument that allows for the very first step for us to be able to mitigate this disease. In terms of validation, the model C statistics is 0.72, which is not perfect, but we consider this as a win, given that there is no precedence. Anything above 0.5 would allow us better discrimination than random chance. In terms of calibration, we identify some problems in the higher predicted probability area that model starts to overestimate, but these can probably be corrected with the further data and identification of different feature. And just to show that this data set in our approach has endured several peer review publications. In terms of cost benefit and opportunity, the cost of app development is about $25,000. The benefit is really this app opens healthcare's to an entirely new patient group that had been previously not treated for this indication. So this is a large population, and considering that the mean hospital revenue that each cardiac open heart surgery case brings, which is $21,000, if this app essentially identifies 2 patients that undergo surgery for this indication, then the app really pays for itself. In terms of the market, we are looking at over 1,000 US hospitals that performs the ascending aortic operation for this specific indications. And the competitive advantage, as I mentioned, is the unique data set and the algorithm that is derived from it. In terms of milestones, app development requires further funding, as well as the algorithm requires validation in a prospective fashion to test this in a different population and whether the yield is actually as predicted. And scaling dissemination also requires guidance. So thank you very much for your time.
Thank you, Dr. Mori. Wonderful presentation, very interesting problem. We have time for a question from a judge.
Thank you for a wonderful presentation. I do have a question about the integration of the application with the EHR. Could you talk a little bit about how you gather data from the electronic health record, or whether or not it's integrated with the EHR at this point?
Yes, thank you for the question. It is not, and we intentionally wanted to restrict the model to very simple parsimonious model because that overcomes the overhead of figuring out the implementation of this into different EHR system, which I think a lot of complex model has encountered in the actual implementation and application to the patient care.
Great. One other follow-up question. In terms of the business model and how you see disseminating this with a larger population, would you be selling directly to hospital systems, or would you be partnering with different EHR groups such as Epic or Cerner? Have you given some thought to that?
I think, well, as it current stands, we are bypassing the EHR system. So I think we are going directly to the hospitals, perhaps in a subscription type of model.
Okay, great. Thank you.
Thank you.
Thanks. Great questions. Wonderful presentation, Dr. Mori, and thank you so much. I'd like to move on to our next pitch, which is Dr. Sadeghi. Dr. Sadeghi, I'd like to invite you to put your slides up. I think you're on mute. Are you on?
Hi again. I'm going to talk about our agents for imaging protease activation in cardiovascular pathology. What's the problem? The tissue remodeling is a common pathological process in cardiology and in other fields of medicine, for example, pulmonary diseases and cancer. Aortic aneurysms and cardiomyopathy are highly prevalent diseases associated with adverse tissue remodeling. Non-invasive tools or biomarkers to track tissue remodeling are lacking. These are necessary for early diagnosis, risk stratification, therapy implementation, and tracking the response to therapy. Just to talk about abdominal aortic aneurysm, it affects 4% of older men and is responsible for 10,000 deaths in the US every year. As you know, we screen these patients by serial, we follow these patients by serial CT and MRI or ultrasound while they're waiting for, when the aneurysm size reaches a certain threshold, they undergo surgery. But many deaths occur during this surveillance period. So we need better tools for aneurysm risk stratification. As many as 1 out of 200 adults suffer from cardiomyopathy. Cardiomyopathy can lead to heart failure, and the mortality after the diagnosis of heart failure is 40 to 60% over 5 years. Current evaluation of cardiomyopathy is based on LV anatomy and physiology. This misses the opportunity to manage the disease based on the biological processes that promote the development of heart failure. So we need better tools to assess the remodeling process in cardiomyopathy. Our solution is novel tracers for molecular imaging of matrix metalloproteinases, as shown here. Matrix metalloproteinases are key mediators of pathological tissue remodeling, and these can be used to better assess the rupture risk in aneurysm, personalize therapy, and track the response to therapy. The US market for PET tracers alone, and as you know, there are also SPECT tracers, reaches $4 billion per year. And we hope to tap into this market and even expand this market by introducing new applications and tracers. We don't have any competition right now because there is no FDA approved tracer for this indication. This is completely novel. The advantages of our approach is that we have strong preclinical data that show the effectiveness of our tracers. For example, in this mouse model where the uptake of our tracer in aneurysm was able to predict those animals that developed the aneurysm or died of aneurysm rupture versus those who didn't develop aneurysm. We have a direct path to clinical translation for our tracers, and we are on our way to perform our first in-human studies. Currently, Yale has an IP on these tracers. Their chemical synthesis has been optimized for medium-scale production. Their biodistribution, molecular targeting, and effectiveness was demonstrated in multiple murine models of aneurysm, and we have secured NIH funding for first in-human studies. Our go-to-market strategy is to de-risk the technology, i.e., performing the highest risk part of this, at least the scientific part of the studies early on ourselves through NIH funding. We're going to perform preclinical toxicology and pharmacokinetics, and large-scale GMP production, IND filing, and again, as I said, we're going to select the lead agent for first-in-human studies. And our plan is to either license this or start a company with these tracers and maybe other tracers developed at Yale for further development and human studies. We have a large team of investigators at Yale and Washington University, consisting of experts in cardiology, vascular surgery, CT surgery, molecular imaging, chemistry, and a lot of experience in taking new tracers to humans. What we miss is a knowledgeable, effective business manager to help us move this forward. Thank you.
Thank you, Dr. Sadeghi. Fantastic, interesting presentation. Do we have time for a question from a judge?
Hi, I'll chime in then. What facilities do you have available to run the necessary animal toxicology studies?
So we are working with, in our grant, we are working with WashU, Washington University in St. Louis, who's done multiple studies like this. And these studies are ongoing right now, in fact. So we're making the tracers, large amounts of that for those studies. So that's in the plans, already in process.
I'd like to ask a quick question as well. For a business manager, do you have a plan for hiring a part-time business manager, or will you be starting a company and offering some kind of ownership or participation in the company you're starting to engage that business manager to help you move this forward?
So I'm open to any suggestion here, but my hope was that Yale would take these and other tracers that are developed at Yale and start a company, hire a business manager, help us hire a business manager to take them forward. So this is a very big untapped essentially resource here by us and other investigators at Yale that can be put together or even by themselves and move ahead. So I don't know if I answered your question, Dan, directly or not.
Absolutely. And I have been turning off my video in between just because of its internet connectivity and whatnot. But thank you so much.
Dan, can I ask a quick question and follow up?
Yes, we have time.
So Mehran, that was wonderful. And I think I guess my question relates to the human studies that you are going to pursue, and whether you mentioned first in man, but I guess are there particular pathologies, and at what stage of that pathology are you going to identify to kind of put this tracer in man and track its efficacy, I guess, or accuracy?
thanks for this question. We had to think about this for 3 years. Eric, and the issue was we wanted to have a tissue, cardiovascular tissue, that we have access to cardiovascular disease, that we have access to tissues. And for this, we focused on aortic aneurysm, where we still, abdominal aortic aneurysm, we harvest the tissues. The application of the tracers extends well beyond abdominal aortic aneurysm or even cardiology. It extends to everything. But we focused in abdominal aortic aneurysm. And our first studies are focused, first of all, in normal subjects to show safety. And the second set is to validate that we are effectively imaging protease activation in aneurysm. So that's the first step, to show that the tracer does what we think it does in the mouse. And also, so after that, the next step would be a multi-center trial to essentially now show the effectiveness, for example, in risk stratification in aneurysms. So, and it could be in heart failure. At that stage, it could be heart failure, it could be pulmonary, fibrosis, it could be any other diseases which are associated with tissue remodeling.
Thank you so much, Dr. Sadeghi, and for our judges. A quick word about timing. We will be going till about 5:09 to allow Dr. Spatz to have her full time. And I would really encourage everybody to continue participating till that time, if at all possible, because it's a very exciting set of presentations ahead. Dr. Sinusas, I'd like to invite you to show your presentation.
Can you see my screen and hear me?
Yes.
For some reason, I can't get it to go full screen.
This is okay.
Okay. So good morning. I'm a co-inventor of a novel catheter-based radiotracer detector system for guiding cardiovascular interventions. I'm presenting this technology on behalf of my collaborators at Yale and Athron, a startup company created for advancement of this technology. Our device addresses an important and unmet clinical need. In the US, there are over 1.5 million annual cases of myocardial infarction, which are often complicated by heart failure and recurrent atrial fibrillation and ventricular tachycardia. Prevalence of heart failure in the US is projected to be 8 million by 2030. And the prevalence of atrial fibrillation by 2030 in the US is 12.1 million. Catheter-based diagnostics for the delivery of therapeutics to the heart is actively performed for the treatment of heart disease. Successful therapy depends on locating the ideal site for delivery of therapy. Current methods for myocardial characterization and anatomic guidance are not adequate, and our novel method is based on the biological characterization of cardiac tissue as defined by uptake of molecularly targeted radiotracers. We propose a disposable steerable catheter for the identification of diseased myocardium for targeting therapy via the detection of systemically delivered targeted radiotracer. The detector system is illustrated, and clinical applications include the guidance of ablation of ganglionic plexus for the treatment of atrial fibrillation, the guidance of ablation of the peri-infarct zone for the treatment of VT, guidance of the delivery of stem cells, gene therapy, or hydrogels, or the guidance of myocardial biopsies for the diagnosis of cardiac sarcoidosis or myocarditis. So on the left is a high-resolution image of a 3D movie of the heart using radiotracers that target sympathetic activity. We observed diffuse activity in the LV and focal activity in the areas of the atria that represent the ganglionic plexi. Studies suggest that the added benefit of ablating these plexi in combination with pulmonary vein isolation for the treatment of recurrent AFib, our detector injector system may locate and ablate these plexi. On the right are FDG PET-CT images of a patient with suspected cardiac sarcoidosis. Focal uptake is seen in the intraventricular septum, in the RV, and the LV free wall. Our catheter can locate areas of local inflammation and can guide myocardial biopsy for the diagnosis of cardiac sarcoidosis or myocarditis. This year, we were issued a patent for this technology. Our disposable catheter is shown with one or multiple beta radiation detectors with the integration of an intramyocardial injection system for guiding intramyocardial delivery of therapeutics. The catheter is an 8 French and incorporates a plastic scintillator and a silicon photomultiplier sensitive to the detection of only local beta radiation emitted from any PET radiopharmaceutical. Our prototype catheter with an integrated injection port and retractable needle are shown. The catheter could also incorporate an ablation device or biopsy forceps and can be positioned over a guide wire. We evaluated a prototype catheter using a pig model of reperfused myocardial infarction. And we injected FDG 1 week post-MI and performed in vivo and ex vivo PET-CT images. And then this was followed by interrogation of the myocardium with our radiation detector. And shown are the FDG PET-CT images of a pig heart with a large lateral wall MI and a section of myocardium containing viable and non-viable myocardium. The catheter provided quantification of radioactivity at the endocardial surface, which correlated with the PET images and tissue counting, and demonstrated both sufficient spatial resolution and temporal efficiency for real-time endocardial mapping of radioactivity. The Atheron was formed to develop and market this technology. We have obtained initial seed investment, have built several prototype devices, and conducted benchtop testing and initial pilot large animal validation studies. Atheron and Yale were awarded the joint patent, and we've licensed 4 other patents related to this technology. There are several companies that have approved catheter delivery systems, including Biosense Webster, Biocardia, and Saliad. Therefore, our device would be predicated on these existing systems. However, none of these companies have molecular guided catheter systems. So what are our next steps? To identify an industrial partner for the development of a steerable catheter system, to identify an investment partner for the translation of our technology to clinic, and to prepare NIH grants for advancement of the technology. Our financial projections are shown on this table. Our goal is to sell 10,000 catheters during year 3 for $1,000 each, with a projected cost of manufacturing of $150 per catheter. We're seeking an initial investment of $15 million over 3 years and a projected gross profit of $90 million over the 5-year period. Thank you.
Wonderful, Dr. Sinusas. We have time definitely for a question from one of our judges.
Hi, do you have the ability at Yale to make one of these catheters and sterilize it so that it would be suitable for a first-in-human clinical use?
So our partner is Atheron, which is a company, a commercial company that develops and sells intraoperative probes for detection of metastatic disease. And so this would be an extension of their surgical technology to a catheter-based technology. So they have manufacturing facilities, but we would need to develop a industrial partner that would help put this technology onto a catheter.
Okay.
Sorry, thank you very much. It's a very, very interesting presentation with some interesting technology. We're going to move on to our final presentation. It's been an absolutely breathtaking morning so far in terms of all the innovation and ideas. It's overwhelming almost. I'd like to invite Dr. Spatz to show her slides and present. And please stay with us if you can for this 5-minute presentation and questions afterwards. And remember, I'll put the slide up for audience participation at the end if you want to rank and vote yourself.
Okay, hi everyone. Thank you for sticking with this and thanks for the opportunity, Dr. Jacoby. I'd like to present Pressure Points, a personalized app for cardiovascular disease prevention on behalf of my team, Dr. Onuma, Dr. Liu, Meadows, and Berg, and with our advisors, Dr. Failin and Dr. Krumholtz. This is a fantastic team that Dr. Onuma and I had an opportunity to work with to submit an AHA SFRN grant last year. While the work was not supported, we remain committed to the mission of the work, which really focuses on one patient, Mr. Green, who is our patient ground zero and who reflects the failures of our healthcare system in cardiovascular disease prevention. Many of you have met Mr. Green before, either in my prior talks or having cared for him in our hospital system. He's a single dad with 2 kids. He works 2 jobs, has little social support, and lives in a community with little access to healthy foods, pharmacies, or safe parks. We first met Mr. Green when he came in for acute heart failure due to poorly controlled hypertension. And it was during this hospitalization that we learned the details of his lives. He had seen our health system twice before in the emergency room and primary care with significantly elevated blood pressures. And once again, we saw him in our cardiology fellows clinic after his hospitalization for heart failure. On all of these visits, we focused on guideline-directed medical therapy, but we lost the critical information about the context of his life that was so critical to medication adherence and to understanding what his life would be like outside of the office and tracking him and being able to stay in touch with him. Ultimately, he presented with a disabling stroke from an LV thrombus. Mr. Green lives within a community of New Haven, which has the highest hospitalization rates for hypertension, accounting for 1% of all emergency department visits. And he lives in a zip code, New Hallville, with the lowest life expectancy in all of Connecticut, under 74 years. Mr. Green has a unique biology, lifestyle, and emotions, and unique stressors, physical activity, and sleep patterns, all influenced by his environment. And we want to connect with the Mr. Greens of our patients, of our patient world. And we want to connect with them in meaningful ways, but it's hard. On the left is more what our visits look like. We're buried in Epic. The outside data that are so critical to understanding our patients are difficult to capture and integrate with biological data. And our current technology stifles connection. On the right is something potentially more reflective of the kinds of conversations that we want to have. We want substantive data, personalized data about biology and biography. We want it integrated and interesting and sticky so that it means something to the patient. But we lack the digital tools to bring this all together. Our solution is to develop those digital tools that would help the Mr. Greens of the world, bringing real-world data from wearables, surveys, and social determinants of health, together with the important information that exists within our electronic health record, such as their risk factors and healthcare utilization, and bringing them together using data science and machine learning algorithms to integrate that data to create meaningful stories of people's lives and use that to direct goals and track patients. Pressure Points is innovative because we do deep phenotyping using multiple data sources and providing new learnings. We have an intentional focus on health disparities built in from the very beginning with the types of data that we're collecting. It's unique in that we're bridging the office, the clinical setting, with the home, the behavioral setting, using dashboards and in the future apps based on gamification. We're unique in that we use rapid prototyping and testing with end users using a design thinking methodology. And we're focused on that last mile, focusing on the needs of all users, patients, clinicians, and health systems, and using implementation science to test the effectiveness of these interventions. What's the problem? The problem is that 40% of Black adults in the United States have hypertension. The problem is that we spend $45 billion per year on hypertension and $2.5 billion on social determinants of health. We have 15 minutes per patient visit, and yet we need to close this 13-year life expectancy gap. So the problems are huge. What's out there? Well, there's lots of digital health apps that are out there. I'm showing you 2 of them that we currently use in our practice, Vivify and Livongo, but I can tell you that they're generic. They're frustratingly impersonal. They're not integrated with medical record data, and they seem to have left out the docs and the patients that we see in the design of their apps. The opportunity is for us to partner with health systems like Yale New Haven Health, who are struggling to meet population health goals and value-based care, to better support clinicians using better data, and to better support communities that would build better partnerships with patients within those communities. We've done a lot of this work preliminarily. We have parts of it. We have a digital hypertension program in which pharmacists meet with patients. We do home blood pressure monitoring. We connect with the medical record. We address social determinants of health, but it's clunky, it's fragmented, and the data are not together or in any kind of clean dashboard that we could use it to engage with patients during our clinical sessions. We're also developing machine learning algorithms to phenotype blood pressure patterns from a grant with the National Institute of Minority Health and Health Disparities. And so we have the data science know-how, but again, this is stuck in the research world and we haven't put it into clinical use. Our ask is for seed money to begin to integrate real-world data with EHR data into an app or a dashboard. This is the first part of our plan. Our resources that are needed are data scientists and healthcare designers. We'd like your mentorship taking this product to market and see this as being continued through support by R01s and venture capitalists. Thank you.
Fantastic. Dr. Spatz, Dr. Churchwell wasn't able to make it this morning, so I'm just going to ask the first question. The product that you are thinking of putting together here seems kind of, it feels diffuse to me. What is the specific product that you would be coming forward with, and how would you interact with health systems to create a financial basis for that product or a market?
So the product is a digital health app that integrates data coming from wearables, survey data, and the electronic health record. So it does have a data feed from the electronic health record. So we would identify specific concrete variables that would be filtered into this data app and put into a dashboard. The idea is that this kind of app would be downloadable and that the pathways for connecting with EHR data those mechanisms would already be built in.
And would health systems purchase this app or invest in it in order to reduce cost or improve quality?
Yes, so health systems are engaged in value-based care programs. They need to improve their outcomes. This app is focused on hypertension control. And so we would specifically be going to health systems to improve their performance metrics in that area.
Okay.
Can I ask a question, Dan?
Yeah, go ahead.
This is fascinating to me, but Erica, could you answer a question? The wearables part is what's troubling me is, are you assuming that in the minority community that they already have the wearables or that they would be provided as part of this intervention? And how would this data flow from the actual patient?
Many patients have wearables or that are built into their phones, such as physical activity and steps. So that data are available to us. Most of the additional wearables would need to be either provided or purchased by the patient.
And do you experience any trouble with getting routine blood pressure monitoring from patients who have presented with extreme hypertension when they go back into the community?
So Medicaid covers regular standard blood pressure monitors, not the Bluetooth monitors. We have a pilot program supported by the Heart and Vascular Center that has provided those Bluetooth monitors to these kinds of patients at a premium cost. So we think that patients are asked to purchase blood pressure cuffs all the time and do so. And so we have not met resistance for the blood pressure cuffs. Yeah.
Okay. Thank you.
Okay. Well, thank you, Erica. It was amazing. This brings us to the official end of part 1 of the Innovation Grand Rounds. I would like to offer a very hearty thank you to our participants. I was amazed by the work that's being done, and the presentations were, to a point, fantastic, very interesting and innovative. I hope that you had a good time in participating and that it wasn't too stressful. I hope that people enjoyed watching, and I hope that you will tune in next week when Dr. Singer and Dr. Cartier will speak to us about practicalities of innovation resources at Yale and Dr. Singer's personal insights with regard to giving your idea a chance. Thank you so much, and wishing everybody a great week. And thank you again to our participants and our judges. Thank you so much for your time and investment in this project.